Yay. Okay, here we go. Hi, I'm Miss Sloan, and this is my honors bio class, and we miss you today. We have a very small review, a small contingent here, but I'm very proud of you for showing up. And this is chapter 24, DNA structure and replication. And then we're going to break up the quiz for chapter 24, and then you'll have biotechnology. That'll be in your next one. All right, I am going to make myself... It's funny how sometimes it makes it big right from the start and sometimes not. Okay, there we go. So here's the code if you just jumped in. NKXFGW. Got it? NKXFGW. Okay. All right. First thing is I would make sure that I knew the history of our understanding of DNA. And so there were some key experiments we went over. All right, and so take a look through those. Make sure you feel comfortable with those. Hershey and Martha Chase, Chargaff, Rosalind, Franklin, and Wilkins, and Watson and Crick. Then what is the structure of DNA, the details of that? What are the steps of DNA replication? Can you describe the central dogma? What are the steps of transcription and translation? And there are no additional objectives for this section of the quiz. Can you tell your bio buddy, what are you feeling comfortable with on this list, or what are you feeling worried about on this list? Have that discussion. Go ahead. They're being very quiet. Are you, that's the code right there. You got her? Okay, thank you, sweetheart. Okay, you good? If there's parts on there that you're really worried about, make sure that I talk about that today in a way that helps you to understand it, all right? So first off, let's talk about Hershey and Chase. Remember, they were trying to figure out, is DNA our actual hereditary material? So they did this little real tricky thing with a virus. They didn't know the structure of a bacteriophage. That's a virus that infects bacteria. But they knew that it took over the bacteria. So they are wondering, what did it overtake it with? So they labeled the protein part of the virus, and they labeled the nucleic acid portion of the virus. So they used P32, and they labeled the nucleic acid, and S35, a sulfur isotope, to label the protein. And then they were wondering, what's going to show up inside? Now, if you remember from that experiment, they gave it enough time to infect the bacteria. And then what did they use to separate the bacteria and the virus from each other? A blender, exactly. And then they used a centrifuge where they spun it really fast, right? And then they looked to see what's inside of the bacteria. Does it have radioactive sulfur? An idea then that protein was what got passed on, or did it have radioactive phosphorus? And what did they find inside the bacteria? It was radioactive phosphorus, right? And we know the structure of DNA. It's a nucleic acid. And what are all nucleic acids built out of? A base, nucleotides, which is a base, sugar, phosphate. So that's why they could tell when they looked at it that the radioactivity from the phosphorus was down there, because that's in fact what the virus had injected. So that was one of the key experiments to show that DNA is our hereditary material. I'm not doing all of them, just some of them, right? And then the next step was, what is the structure of DNA? So there was a man named Chargaff. And Chargaff, in his research, he came up with I don't know where to stick myself here. He came up with two items. He said that, number one, that the adenines, thymines, and guanines and cytosines are specific to each species, but within a species, what did he demonstrate? A's and T's were the same number percentage, and the C's and G's. We now know because there's complementary base pairing. He didn't know that at the time. Okay? So then you want to be prepared if you have a question where if I give you one of the percentages of just one of the nitrogenous bases, that you could figure out all the rest. So why don't you try to do that real quick? And if you're at home, try and do that at home. That is exactly the type of question I tend to ask on a quiz. We good? Who does thymine base pair with in DNA? Adenine. So if thymine is 10%, then adenine has to be 10%. 10%. So together, those two percentages equal how much? 20%. So how much would I have left over then? 80%. And that would have to be shared between guanine and cytosine. Okay, ask the person next to you. Do you understand that for sure? Do you get that? Okay, so it shouldn't matter what I give you, you can figure it out. So Watson and Crick are the ones that came up with that. So they used Chargaff's information to help them with their thinking. But there was another experiment as well. Okay, so here we have Rosalind, Franklin, and Maurice Wilkins. And what they did is they would take crystallized DNA and they would shine an X.ray through it, like you would take an X.ray of a broken bone. And then from the pattern, because they'd done this a lot with a lot of other chemicals, they knew what this meant. And they knew that this was a double helix. There was a repeating pattern in here of constant diameter. So now, I always give you the analogy like you're looking down a what? A spiral staircase, right? And you're looking at those steps going around. We now know those steps are composed of nitrogenous bases held together by what kind of bonds? Hydrogen bonds. And the side of our ladder is what? What did you say? Yeah, it's covalent bonds, but what's the edge of our ladder made out of? Phosphates and sugars, alternating phosphates and sugars. So Watson and Crick used all of that data in order to come up with their structure of the double helix. Okay, so you have a sugar phosphate backbone, okay, and then you have paired bases held together by hydrogen bonds. All the other bonds are not hydrogen bonds, they are what kind of bonds? Covalent bonds. Okay, look at your person that you're sitting next to right now. Are you good? Any questions so far? Are you feeling strong? All right. So then I have a compilation slide and of all the things you can review with DNA and DNA structure. And this harkens back to when we were talking about the 4 important organic molecules. So hopefully this part is a review. So we know that all nucleotides, and I took this right out of that presentation, right? All nucleic acids are built out of nucleotides, and a nucleotide is a nitrogenous base, a sugar, and a phosphate. The sugar in DNA is deoxyribose. The sugar in RNA is ribose. Good. We know that DNA is double-stranded, whereas RNA is single-stranded. Good. We know that adenine and guanine are both referred to as purines because those two nitrogenous bases have 2 rings to them. So you've got 2 carbon rings held together, whereas thymine and cytosine, and what's the other base found in RNA that's not here in this picture? Uracil, those are all pyrimidines, right? And pyrimidines all have 1 ring. And the way the DNA keeps its constant diameter of 2 nanometers is you always have a purine bound to a pyrimidine, right? And the purine could be over here and the pyrimidine here, or the purine here and the pyrimidine. It doesn't matter because you're basically going to have a total of 3 rings going across, right? With a purine and a pyrimidine. All right, and then let's see. And then it's 2 chains of nucleotides held together by hydrogen bonds. Now, the part that we focused on, though, was that numbering, right? That was the part that was new. And that's because when we look at a sugar, okay, when you look at that sugar, even right here, you can see the numbering, right? Carbon 1 is always the carbon in that sugar that's bound to the nitrogenous base. And if you wanted to go up, if you see this as a house, if you wanted to go up on the house, what do you have right there on the roof of the house? Oh, well, the chimney, I say this is the chimney over here, but what's right here? That's oxygen, right? So you can't go up that way. Instead, you need to go down. So when you go down, that's why this is carbon number 2, 3, 4, chimney makes 5. And I always think like a puff of smoke, that phosphate is coming out the top. Thank you, Nikki. All right, but when you look across the street at the other strand of nucleotides, all the houses are going in the opposite direction, right? Just like the street out here on Canaan, the cars are going two different directions, right? That's called being anti-parallel. So when we look at those sugars across the street, carbon number 1 is still the same. It's the one that is connected to the nitrogenous base. But now this house is upside down. I still can't go here because there's an oxygen there. They haven't drawn it in this picture, but you can see it over here, right? You can see the oxygen. Okay, so that means I need to go here for 2, 3, 4, chimney makes 5, puff of smoke. So my house is upside down. When this becomes important is when we do things like DNA replication and we do transcription. Remember when we talk about the leading and the lagging strand? And that's because our enzymes are very, very what? Specific, right? So they bind to, what do enzymes bind to? They're what? Substrate. So if the sugar is going a different way, it's not going to fit, right? And so that's when it becomes really important. So when you look at a strand of DNA, when I look at this all the way over to the right, I know this is carbon 1. I know this is carbon 1 because it's connected to the nitrogenous base. 2, 3, 4, 5, puff of smoke. This is the 5' end. And if this is up, then this has to be down, right? This has to be the 3' and vice versa, 5 and 3. Check to the person next to you. See if they're feeling comfortable with that. If you need me to go over that at all anymore, I will do that if you need it. Okay, and you can see that numbering right here too, right? In the very first one. Can you see the numbering? Okay, anything you want to ask me about that? Okay. So when we talk about DNA replication, it's called semi-conservative replication because the old strand here in blue serves as a template, bless you, for each of the new strands here in red. Okay, so that's called semi-conservative. Conservative would be if the old one served as template to build the new, and then the two olds got together and the two news got together. Okay, but semi-conservative, you got an old and a new. Original parent strand serves as the template. Okay, the enzymes that are involved in that, right? I taught you that you don't have to know this one, but I did teach you topoisomerase, right? That undoes the superclone. Then we said we got to get this DNA to unwind and unzip. What did we call that? Helicase, right? Now we have two strands. We have a leading and we have a lagging strand, okay? So our leading strand reads from 3 to 5, and our lagging strand reads the opposite, 5 to 3. And there is a primer to get the party started, but I don't hold you accountable for that. The first, besides helicase, you need to know DNA polymerase 3. And it, boom, boom, boom. On the leading strand, real quick, easy to read, right direction. If there's an A, it brings in a T, G, C, C, G, etc. The lagging strand, it can't read this, right? It's not in the right direction. So it has to jump back and then backstitch, jump back. And that's why we go boom, boom, boom. Okay? Now what we have created, I need like 2 more arms here. These will twist up and be what? Sister chromatids held together by a centromere, and they will separate during anaphase, right? So the analogy I always give is if DNA, right, is your code book for life, right? If I'm going to make another whole biology classroom, they're going to need another book, right? So the whole book is replicated. These are your sister chromatids, and they are made during the S stage of interphase, and they separate during anaphase. Okay, you all right? Okay. So then this is showing you that here. One thing, because I'm pretty sure I taught you Okazaki fragments, yes? So that's because the lagging strand is made in those short segments. Those short, it's 1,000 to 2,000 base pairs, but those are called Okazaki fragments. Did we do ligase? Okay, I couldn't remember if I said that, but I said it. Okay, good. All right, and then here, I just listed off all of those enzymes, which I'm blocking. Okay, just listed them off: DNA helicase, DNA polymerase, and DNA ligase. Those are the ones that I want you to be able to know if I asked you about them on a quiz. Questions, concerns? We're okay? All right. So then the next thing I want to go over with you is the central dogma. Maybe baby. Let's go. Please don't be bad. Please don't freeze. It's freezing. Is it freezing on your screen? Let's see. Page is unresponsive. Oh, wait. I think it's moving. Is it moving for you? Okay. It was telling me page is unresponsive. I'm like, no. Okay. So, yeah, you can drag the dots. The central dogma is talking about the flow of information. So we know the code book right here, making more code books, like another whole book, that's called DNA replication. Where does DNA replication occur? Inside the, if you're a eukaryotic cell, inside the nucleus. Good. And then transcription is, you know, if I'm in the kidney, I just need kidney information. I don't need the whole book. I don't need to know how to build hair. I don't need to know how to build a heart. I just need kidney information. So that's when you go in and you make a copy of the kidney information. Okay, here's my copy. So when I have that, this is taking a message from the book out into the cell where I have the factory in order to build the protein that I need, right? So what would I call this? It has the mRNA. It has the message. Where did it get the message from? DNA. Then this mRNA goes out and gets on the workbench. What's the workbench called? Ribosome. And there I want to build my protein. Who's going to bring me the protein components? What are proteins built out of? Amino acids, so tRNA. And it's going to transfer it to the workbench so I can start stitching those amino acids together to build a protein. Okay? The workbench, a ribosome, is made out of two things. What are the two things a ribosome is made out of? rRNA and proteins. That's what builds that bench. tRNA is made out of RNA, okay, and it transfers an amino acid. So transcription is using the DNA to make your RNA. Translation, I'm putting it into a new language. Instead of the nucleic acid language of the previous two steps, I'm putting it into a protein language. Okay, look at your bio buddy, see if you're okay, surviving. Okay. All right, so it's now when I wanted to kind of compare and contrast DNA with RNA, DNA is double, RNA is single. DNA has the sugar what? DNA has the sugar thymine, and RNA has the sugar. No. Let me say that again. DNA has the sugar deoxyribose, and RNA has the sugar ribose. It's okay. I accepted it the first time. I'm tired too. And then DNA has the base thymine, and RNA has the base uracil. Okay. And we have three kinds of RNA, right? M, T, and rRNA. There's actually more RNAs than that, but those are the only ones I'm holding you accountable for. All right. You okay with that? Okay. So in the process of, come on now. Yeah. So this is just showing you the three kinds of RNAs and what each does, right? Messenger RNA carries the instructions from the nucleus out to the workbench. The workbench is is a ribosome made out of rRNA and proteins. And then who's going to bring us our protein components? tRNA. Now, every three bases on the mRNA is called a what? Codon. And that is complementary to a that's on the tRNA. Okay, they're complementary to each other. So that chart, remember, we use the codons to look it up, right? The codon chart, we use the mRNA. And so if I was going to do replication, right, I do the complementary base pairing for DNA. You'll see thymines in there. But if I'm making RNA, and that's what that would be like a really typical question is I might say the DNA is this, what's the mRNA? Or the mRNA is this, what would the tRNA be, or what would the DNA be? Okay, so that's what you want to be able to do. So When you go from DNA to mRNA, you're just putting the complementary base pairing, but wherever you want to put a T, you're going to put a U, okay? And then once you have the mRNA, when you're trying to figure out the tRNA, remember to keep the U, right? Keep the U instead of a T. And then the transfer RNA is carrying the amino acids. How do we find out what amino acid? We look up the codon. That's a codon chart. Okay, so you look up the codon. Good, you're all right? Okay. And then, so this is showing you, let's do that song for that part. So the DNA unwinds and unzips, helicase. We have a leading and a lagging. Put the lagging strand away. Now we're going to get what? RNA polymerase binds to the promoter. Boom, boom, boom. Make your mRNA. And this is A with U, G with C, right? All that. Okay. Boom, boom, boom. Make your mRNA. Peel it off. Set it over there. Go get your DNA. Bring it back. Zip it up. Put it away. Here's your mRNA. Now, this mRNA has to be processed, right? If we're in a eukaryote, you have to put on a cap, 7-methylguanosine. You don't need to memorize that. And a tail, 100 to 200 adenines. And then you cut out the introns before it leaves the nucleus. When you cut out the introns, what are you leaving behind? Good. So we can see that here. Okay, we can see the RNA polymerase is the big purple thing. I can see the DNA in the background. Here comes the mRNA. It's got a cap. It's got the tail. What color are the exons in this picture? Can you tell? Orange, yeah. And what color are the introns that we're cutting out? And we're going to be careful of that because it's counterintuitive. Like in, we think keep in, right? The way we remember that is the exons get expressed. Good. And then we go out and get on a ribosome. Ribosome is our workbench. So I think I have one more slide here about cutting out the introns. But I could also cut out one of these exons if I wanted to, right? So that's why your DNA can give you so much variety in your RNA, because what you cut out. Then the steps. The steps, first the mRNA has to hook up with the small ribosomal subunit. So small ribosomal mRNA. Okay. Every 3 bases is called a. The first tRNA lands in the P spot. Okay, the first tRNA lands in the P spot. The second tRNA lands in the A spot, right? But as soon as the first tRNA lands, it's like you lock it down, like this is what we're going to do. And so the large ribosomal subunit settles down. Okay, then your next tRNA lands and the new guy always gets dumped on. So the ribosome shifts and shifts the amino acid onto the new guy. And now the new guy is in the P spot and the old guy is in the E spot for exit, right? Now the A spot's opened up again, another tRNA and shift, another tRNA shift, 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 shift till we reach a stop codon. And then everything kind of breaks apart and you have a chain of amino acids. And that mRNA could be used again and again and again, making more and more proteins until it's degraded. Right. And I had another picture for that. And ask your bio buddy, is there any questions you have about that? Feeling good? Okay. Next, mutations. So mutations are any permanent change in the DNA. So that could happen like accidentally when you're doing your boom boom and you're replicating, but that's very rare, but it could. Mutagens, chemicals could alter your DNA, sunlight, exposure, smoking, any of those things. That could change. And then transposomes. Remember transposomes? Those are those jumping genes. They literally jump out of your DNA and move to a new location. Evidence of transposomes are the Indian form. Okay, then we talked about 2 kinds of mutations in particular. One was a point mutation. So I could point to the nucleotide that's an error in that one, a single nucleotide, wrong. Okay, and in this mutation, it was called a what? Silent mutation. Why? It still got me ultimately the same amino acid. And that's because there's redundancy in our code, and there's like more than one, not for everything, but more than one way to get, let's say, proline. Okay, this is a nonsense mutation because bummer of all bummer, right? 3 of our 64 anticodon, or 3 of our 64 codons are what? Stop. So you happen to mutate into a stop codon. So midway through the building of your protein, it's stopped. It's like a house that gets, it gets framed up and it gets the plumbing and then they never finish it. Okay, can't live in it, can't use it. And then in this case, the first one was changed. That's always a bad one to change. You get a totally different amino acid, which means it doesn't probably fold into the right shape, and that is called a missense mutation. Is there anything worse than a point mutation? What's worse? You remember what it is? Frameshift. Yeah. A frameshift mutation is where you get a deletion. So it throws off your reading frame, right? Your first codon is changed. Or you add in a nitrogenous base and it throws them all off, because then every one of these is going to be wrong. And so we just use 3-letter words to represent codons because we could understand that. Questions about frameshift? Okay, guys, then that was it. Do you got it? I am so proud of you for coming in on your Friday afternoon, and I'm sure people will thank you, because if you weren't here, this would not be recorded. Okay, so good job and have great weekends. You are so welcome.